All right, welcome back, everybody. Just give people a few minutes to roll in. Hopefully your brains are still functional after 2¼ hours of molecular biology this morning. It's not taking genetics over the summer is not a trivial undertaking. That's for sure. All right, so I figured actually it might be kind of better to put this into context, right? So I'm going to my trusty whiteboard up. So essentially, what we're doing is part of this sequence. So we've generated mutation. We have phenotype. And that's basically our flies. If I could do I don't actually have a yellow pen. If I could draw a yellow fly, I would. Actually, let's put that over here. And then we want to figure out what's the basis at the molecular level of that phenotype. Right, so then essentially what we do is we take these flies, we kill them, to extract DNA, right? Because that's obviously where the information that changed to cause this phenotype exists. Right, so that's what we've got to start with to try and figure out what happened here to generate this phenotype. But once we get that DNA out, it's going to be really hard to figure out what that change is because we have to determine the sequence of this fly's DNA or of a particular gene if we suspect a particular gene involved to figure out what that mutation is. And so even after we've extracted DNA, we've cleaned it up, we've purified it, we've got good, clean, intact DNA, we still can't do anything with that DNA just yet. What we need to do is we need to amplify the sequence or the bit of DNA we're interested in so we can get enough of it to be able to do something with it. So the next step, which we're going to talk about today, is about amplifying the target DNA. Now, if this was a blind fishing expedition, we'd have to sequence the whole fly genome, or narrow it down to a region on a particular chromosome by doing the kind of linkage experiments that we talked about earlier on in the course. To kind of restrict it to, I don't know, 3 or 4 potential genes, let's say, right? By doing that kind of mapping experiment that you've just done as part of the Fly linkage report. But either way, we're going to have to amplify that DNA to be able to sequence it. So that's what we're going to talk about today. The next steps would be, and apologize for the writing, would be to sequence that DNA. And that will tell us what that changes that generated this phenotype. So this is the kind of thought process or workflow or kind of, concept that we're working through. Essentially, with the linkage report, we generated the mutation, had a phenotype, figured out where that gene was, narrowed it down to the point where we can actually start working with that sequence. But we need to get from the DNA, the actual genomic DNA of the organism, to our sequence data. Like, what is the sequence of that gene and how did it change via this mutation to generate this phenotype? And so the way we get enough DNA to do that is we do PCR, or also known as polymerase chain reaction. And really, PCR is hard to overstate how important the development of PCR as a technique was. And in many ways, this is actually what I've built, at least my research career on, was PCR, right? So molecular biology, that's kind of how I got my research position at in Columbia, how I got my PhD, and then postdocs from there onwards. So PCR is a super, super, super cool technique, and really the aim of it is you specify which bit of DNA you want amplified. And then it's basically a series of rounds of DNA replication, but only of the particular bit. I'm actually going to draw this out. We've got a PowerPoint, which I can run through too, but I prefer using a whiteboard. I find it just is a little easier. Hopefully you'll be able to see it well enough. If you don't, just give me a shout and I'll zoom in. This is super high technology, right? So the zoom is me picking up my laptop, moving it closer to the whiteboard. Fun stuff. So there's a bunch of different things you need for PCR to work. But the principles are, actually, let's do this with color. Color's always good. So we have our DNA and we have our target, the bit of DNA that we want to amplify. Right, so this is going to be part of the whole chromosome, one of many chromosomes in that organism. So one particular region of that organism's genome you want to amplify. So we have our target sequence. Now to amplify that bit, we're going to need primers. And those primers have to recognize target. Right, and they recognize it by base pairing with the DNA either side of the target. And we always draw them as like these little arrows. What else we need? We need, let's cover this one up quickly. Taq polymerase. And this is, actually, this is the discovery that made PCR work. So PCR was working before the discovery of this particular organism. Does anybody actually know what Taq stands for? Now, if I told you it stands for Thermus aquaticus, which is the name of the bacterial species it was isolated from, what kind of environment do you think Thermus aquaticus lives in?
The deep sea.
The sea, yes. Not quite right on the deep part though, although others have. Yeah, dipper, exactly. Thermus, it's a heat-loving, water-living bacteria. Does anybody know which national park it was found in?
Yellowstone?
Yeah, Yellowstone, exactly. I actually made a pilgrimage to Yellowstone specifically to visit the pond or the hot spring where Taq was found. So Taq polymerase or Thermus aquaticus, the organism happily lives at 70 degrees, 80 degrees Celsius. So the DNA polymerase, which it uses to amplify its DNA or replicate its DNA, also works at that high temperature. That was the key to unlocking the potential of PCR. And then where'd the black pen go? Here we go.
Just a curious question, but
Mhm.
why is it always that in science it's measured in Celsius rather than Fahrenheit? I think I know, but I can't remember.
Because Fahrenheit absolutely sucks as a unit of measurement. That's why.
Okay.
That's the non-scientific version. Scientific version is that Celsius is defined by the temperature of the freezing and boiling temperature of water, which is universal wherever on Earth you are, as long as you're at sea level. Obviously, it changes when you go up or down. Typically, the temperature goes up as you go down, and it goes down as you go up. So, boiling temperature water is like 70, no, something like 85 or thereabouts Celsius on the top of Everest, for example. So yeah, that's one reason. Also, Fahrenheit is a kind of it's not a perfectly linear measurement, I think, is the other one. It's just better. There you go. Metric is typically more sensible. Believe it or not, designed by the French. Much as I hate to admit it, the French actually did something good. Sorry, probably shouldn't say that on. Just betraying my ancestral hatred of the French being a Brit. I can't help myself. It's just handed down through generations. But yeah, I'm not sure about because Celsius depends on melting and boiling of water, but there's actually a kilogram in the Louvre Museum in Paris, which is the original reference kilogram for weight, which is kind of neat. It's so accurate that it becomes less accurate over time because of ablation. It's held in a vacuum, but atoms leave it slowly over time and it becomes less accurate as a measure, which I think is kind of neat. And we need nucleotides, colloquially called DNTPs. Oh, and also some magnesium in the buffer. Those are essentially the components that we need for PCR. And it's a little bit like cooking. It's kind of like magic actually too, because you can't see any of this stuff happening. You just have to assume it does. But we need first and all to design our primers so they match our target and nowhere else. Because we don't want to amplify any other bits of DNA other than our target. That's kind of that's the key. And so what we do is we start with this all as a mix. Typically, actually, we start with all of this as a mix. And usually if you're doing more than one PCR, you'll just make up what's called a master mix, which is all of that all together. And then you just aliquot out the individual reactions, just because it makes it a little bit more accurate and easier to do, saves time. So PCR works by going through a series of cycles, and there are 3 steps to every cycle. So the first step is denaturation, and that's usually at around 98 degrees Celsius, whatever that is in Fahrenheit. I have no idea. And so what purpose this is to generate single-stranded DNA, because otherwise our primers are not going to be able to bind to that DNA, right? So we have our target there still. Then we have, I'm going to get down here, primer annealing. And annealing means different things depending on whether you're working with metal or DNA. But in the case of DNA, what that means is we want our primers to stick. So we're going to have a primer that recognizes one strand, like so. And a primer, I'm really glad I didn't do any more washing today. Looks like it's going to hoof it down. That recognizes the other strand. And so you can see what that generates is it's going to generate like their bookends almost, with the bit of target DNA in between. And then we have extension. So that's at 72 degrees Celsius, because that's the temperature at TAC works really well at. Forgot to say primer annealing, the temperature you do this at depends on the sequence of the primers. So remember we were talking about how many hydrogen bonds you have between A's and T's and G's and C's. So the longer the bit of DNA that makes up your primer. So this is like the RNA primer in DNA replication, but it's made out of DNA. but also the GC content determines at what point these will stick to DNA and at what point they fall off. So you want your annealing temperature to be high enough so they stick only to where they're supposed to, right? The maximum number of hydrogen bonds possible, but not so high that they fall off, because obviously that wouldn't be any use to anybody, right? And so that's typically 55 to 60 degrees Celsius by and large, but again, depends on the primer sequence. And extension,
Okay, so,
go on.
sorry. Just a quick summary, or if you could make it more clear. So the denaturization is where the temperature increases, or?
So that's at a really high temperature, and that's the temperature which DNA falls to pieces.
Okay.
So the hydrogen bonds that kind of hold together the two strands, those bonds break at just below boiling point.
And then during the annealing portion is where that temperature drops, I'm assuming?
Yep, it drops. That's why we use something called a thermocycler, right? Because it changes the temperature. Drops the temperature to the point where these bits of DNA, the primers, are able to stick to their target.
Okay. And then extension obviously goes up.
Yeah, so extension then goes up again to around 72, and our tack basically extends our primers just like it would if it were replicating DNA. Right, so there's the tack. Whoosh. Like so. So that's one round of PCR. Melt, anneal, extend. And then we just do a whole bunch more. So after our first round, we do a second round, and a third round, and a fourth, and so on and so forth. So I'll show you why. Does that make sense?
Yes.
And let me just label these. These are the primers. So basically, after one round of PCR, you've gone from having one copy, which is your original template, and you've gone through the whole process, and now you have two copies of your target. So now, let's say we go through this again. Right. Now we have, let's kind of move it down a little bit. That blue makes such a mess. Now we have 4 bits of DNA. So this is this would be round 2 or cycle 2. Sorry, I just listen over here in my family upstairs. All right, so now we have 4 templates, from our 2 copies, each single-stranded. Now we have our primers will anneal to those copies. Right. And then through extension, we will get now 4 copies of our target. So we're going to have one from the original strand, the template that was there. We're going to have another one from the original copy, another one from the original copy, and a 4th one. Oops, that's a terrible line. Look at that. That wasn't much better. We'll get a 4th one from our other original template strand. So at the end of round 2, we now have 4 copies of our target. We do this again, end of round 3, we'd have 8 copies. 16, 32, 64, 128, 256, 512, 1024, which is after, I should have kept count, 5 or 6 rounds of PCR. And so PCR is, at least at the beginning, an exponential increase in the quantity of DNA. And this is actually the process which allows both anthropologists and forensic scientists to take a really small amount of DNA. We're talking like a little bit of the bone marrow of, Ötzi the Iceman, or a little bit of skin scrapings underneath the fingernails of a victim. And they can take that tiny amount of DNA, or even a swab, right, of someone's nose passages, or mouth that's doing a COVID test. That allows you to take that small amount of DNA that you started with and amplify it into a huge amount of DNA. Because every single round of PCR, at least at the beginning, doubles the amount of DNA that you have present. So if you look at like so, amount of DNA, whatever units that is, and this is cycle number, essentially it takes an exponential curve. Each round that you do, and we do typically it's like 35 cycles or 35 rounds of PCR, you'll get a doubling. And this will only tail off once essentially the Taq polymerase starts getting tired, because you're stressing it out constantly by putting it at 98 degrees Celsius, or you start to run out of primers or nucleotides or whatever, and then this will kind of slow down and plateau. And so this is the exponential phase. And that's where it doubles every cycle. If I can spell cycle. And this is our plateau. Similar to bacterial growth curves as well. When you start running out of materials or start kind of filling your environment with waste, then the growth curve will tail off. But this can be literally as little as 1 copy of DNA. Obviously, you don't want to assume that there's going to be such a small amount. This is typically, you're looking around 35. This is around 5 cycles, give or take. Right. But in theory, as long as you have a single copy of your template, the target that you are after, you will be able to amplify that so you have literally millions of copies of that template. And so now, and if you've all looked at the DNA QC folder with the gel picture, those really faint bands on either side of the molecular weight ladder that was the genomic DNA, you can see how little DNA that you have there. You look at the picture with the PCR run on it, which we'll do for the cleanup and QC on Tuesday, you can see the difference. It's just gangbusters amount of DNA. And so now we've gone from having a small amount of template extracted from a tissue or sample or bone found in a cave in China or something, and now we have enough DNA to actually do something with. That's really the power of PCR. It's super awesome. Questions? But again, this is the technology that underpins kind of every molecular technique that we use, either in police work, in looking at our evolutionary origins, pharmaceutical business. This was how those COVID was sequenced essentially. It's converted from RNA into DNA, and then from that DNA was amplified by PCR. So all of everything that we kind of know about in terms of modern biology and pharmaceuticals and health and the like, all comes from PCR. In fact, if there are any diabetics amongst you, and there most likely is one at least, maybe not in this class, but I've had plenty of diabetics in other classes. The insulin that you would take would be that from the first gene ever cloned for production was insulin, so that people didn't have to rely on insulin that was extracted from pigs. Not only is that great for pigs, because they probably don't want to give up their pancreases just for nothing, but it also means there's less risk of kind of zoonotic disease transfer, because we're very similar to pigs. So if piggies have influenza, then that influenza can probably infect us. So anyway, that's kind of like a kind of cool aside, but it's underpins everything essentially. And let me just see if there's anything in the PowerPoint that I forgot to add. Not really, covered most things. It's always just good as a reminder, because the assignment is based off of what I just said and also what's in the PowerPoint. And what was the name of the lady that is? That's it. Kary Mullis was the researcher that was awarded Nobel Prize for this because of how groundbreaking it was. Adam or Richard, what's your question?
Yeah, my question is going to the lab report document, not the report, but lab document. So I know that you are, I'm assuming, wanting us to find the expected product size
Oh,
and BP.
I'll get to that in a second.
Okay, all right.
So that's the general, good question though. I hadn't actually forgotten. I was just going to get to that, promise. That's the general approach, right? So what we now need to talk about is how does that pertain to what, and good word, by the way, Richard, what we're doing, right? And so we're pretty sure, right, that the change which resulted in our flies becoming yellow was in the yellow gene. Oh, no great surprise, right? Which is a dopamine converting enzyme, right? And amazingly, it's kind of surprises me too. Lots of neurotransmitters are also involved in pigment production, of which dopamine is one of them. I've actually come across other examples too. So if you get rid of neurotransmitter production, you often end up with loss of pigmentation. in animals, which is kind of weird. Don't really understand why that is. But anyway, so what we want to do is we want to amplify part of the yellow gene to figure out what's the molecular change that results in yellow flies. Because clearly something is getting rid of the function of the yellow protein, right? We don't know whether it's a change in the amino acid sequence, right, which results in a non-functional protein. We don't know if it's a change in the regulation of that gene or translation of that gene, right? So we want to figure out what's the basis for the yellow phenotype, molecular level. So the yellow gene, I can't actually remember how many. Oh, I should have done enough times by now. I can't remember how many exons the yellow gene has. 5, I think. Looks something like that, give or take. Here's our transcription start site. So this is exon 1, 2, 3, 4, and 5. Right, and so based on what we already know, and don't forget there's also an AUG just there, that's our start codon, it's got to be at the beginning of our amino acid sequence. We think the problem is around here, right? And so what we've done is we've designed primers to just before and just after exon 1. And so what that will produce is essentially, if we kind of draw it out, a small chunk of DNA, and that is exactly 401 base pairs long. And so what we do is we amplify this using these primers. That will generate a whole load of DNA, 401 base pairs long, which covers the start codon, exon 1, and part of intron 1. And then we'll sequence that. And obviously analyze the sequences. So we can figure out what exactly it is that's causing the change in the protein that results in those flies becoming yellow. So that should also help you answer the questions in the lab.
So our analysis would be the 401 base pairs, correct?
That is correct.
And then the sequences, do we just count however many, I don't know. I was gonna say AUGs, but I don't see U's on here.
For the sequence analysis, that's what we'd be doing in the Eugene lab, which is, I think that's got to be next week. Yeah, so that's going to be, crikey, it's coming to an end quickly. Next week, we'll be looking at the sequences using a piece of bioinformatics software called Eugene, which is going to be next Tuesday and Thursday. And so that's a very powerful piece of software. It's not super intuitive to use, but we'll work through how you would take the sequences that you generate by PCR and then you send off for sequencing. And we'll talk about how you do sequencing too, because that's an interesting process too. And then the sequence reads you get back, we'll be analyzing using Eugene. That'll be next week. That help?
Yes.
Yeah, because the sequence reads are basically just a string of letters, right? So it's going to be very hard to just look at that and figure out what change happened. You have to compare the sequences that we generate of the DNA that we amplify. You have to compare those with what we know is normal, right? So what's the normal yellow gene look like? What do our sequences look like? Is there a difference between the two? And can that difference explain why those flies are yellow?
So since we already determined that 401 base pairs were found in our AUG, how did you come up with 401 again?
So this is our PCR product. And it's 401 base pairs because these primers up here, sorry, I kind of drew them a little small. It's the downsides of having a small whiteboard, right? So these primers we designed based on what we know about the yellow genomic sequence of the gene itself. We designed them so that they would bind to the DNA on either side of exon 1. So when we use those primers to do the PCR, like I showed you in the kind of previous couple of whiteboards, that will generate a whole ton of DNA that is 401 base pairs long, which contains the information in exon 1 and a little bit on either side. So essentially what we've done is we've taken this whole gene sequence and we said, we don't need to know about what's going along over here because we don't think that's involved in our phenotype. We care about what's in exon 1. And so we're just going to amplify a bit of exon, well, all of exon 1, and then we're going to take that amplified DNA and then we'll sequence it. So we can get the sequence of the bit of DNA that lies between these two primers. So whenever you design a PCR, you have to design, well, where are my primers going to bind? What bit of that DNA do I want to amplify? If we wanted to amplify exon 3, we'd design primers that would be either side of exon 3. But we're interested in exon 1, so that's where we design the primers 2 that will amplify that.
Okay, so for some reason it's still not clicking. Something tells me that you have to have some sort of math in there to calculate the base pair, but I'm still a bit confused.
Not really. So, like for example, if our, here's my blue pen. I hate using blue pen. It makes so much mess. Let's say we had a bit of DNA which was ACC, GCTAC. Right, so that's our target DNA. There's going to be a matching strand down here, which will be T, G, G, C, A, T, G. And that kind of continues either way. This is just a little bit of sequence, right, of that DNA. So say we wanted to, our bit of DNA of interest kind of lies over there. So first we'll have to design a primer that would match this piece of DNA. Right, so, oh, could use my purple pen. We would design our primer to have a matching sequence. Actually, up to the bottom one, sorry. So when that primer bound by our hydrogen base pair, hydrogen bonding, base pair complementarity. That primer would serve as a basis for DNA replication of that strand. Does that make sense?
Yeah, I'm just writing it down. If you could tilt it down a little bit more.
Like so?
Or sorry, move it back.
Let me know when you're done, because I'll take a picture of it.
So for the bottom sequence of DNA, you're just matching those base pairs to the ones on top, correct?
Correct. That's exactly what it is.
And that would be T double G C G A T G.
Well, it depends, because the primer is only going to be a short piece of DNA, which is like 20, 22 nucleotides long. But it has to match the complementary strand, right? The bottom strand, if we're going to go that way.
Okay.
So let me just write that down, so otherwise I'll forget. Okay, so let's say that's CCG. I'm helping my pen worked. Okay, so let's say we got rid of, let me get this. So over this end, we have our bottom strand is, what was that? TGG, CATG. And our top strand is ACCGTAC. Right. And our primer at the bottom is going to be, oh, wrong color. Like so. Now over here, we're going to have obviously a different part of the sequence. And I'm not as flexible as I used to be. Actually, I was never very flexible. But so this is GG, TAA, GCT. And so this bit in between is the bit of DNA that you want to amplify, right? That's your target, whatever that DNA may contain. In our case, it's going to contain exon 1. It might contain other bits, depending on what it is you're interested in amplifying. So now you need to have a reverse primer. So this is your forward primer. Because it's going to go that away. Now you need the reverse primer to be able to kind of bookend your target sequence. So let's say that will be CGAATG. So once these have bound to the DNA in the annealing step, when you raise the temperature for the extension step, now the Taq polymerase is going to go, ooh, look at that. We've got template DNA and we have a 3' OH with which, let me just stop sharing so this is bigger. There you go. With which to extend this strand that we're copying. And on the other strand, we have our reverse primer. And that will serve as the primer or the start point for copying the top strand. And so once you've gone through one round of DNA, you now have double-stranded DNA just for this bit and double-stranded DNA for this bit at the bottom, which can then serve as template for the next round, and the primers will bind to the same spots again on the new template DNA, and you'll get extension again, and you'll have more templates for the primers to bind to in the third round. So the primers specify which bit of DNA you're going to be amplifying. And you design them based on the sequence that you know for that gene. And if you don't have a sequence, you just guess, basically, best as you can.
And then, so like with the example that we have for the lab, the GTCC, so basically we rewrite a brand new strand from 3 to 5, and then
And then the same.
Complementary primers.
So once you've gone through full round of, now let me take a picture of this so I can modify it. So once you've gone through the extension phase, right, of PCR, you'll have a matching piece of DNA which looks exactly like the previous bit. But now you have two copies of it. Oops. I was going to introduce a mutation there. That would be good. So now you have, and these are, you know, double-stranded DNA, right? This is new DNA now. And now you have two copies. So when you go to the denaturation step, this double-stranded bit of DNA separates, can be template for the primers to bind to and DNA extension. This strand, or this copy, two strands separates. Each one is now a target for primer to bind to, as these were, and you'll get extension, and now you'll have four copies, four double-stranded bits of DNA. So the primers serve to prime the DNA replication by providing a 3' OH for DNA polymerase, Taq polymerase, to extend the DNA with that way and coming back too. So that's the purpose of the primers in PCR. Does that make a little more sense?
Yes, I understand like drawing it and mapping it out, but it's still that 401 that's really bothering me.
Oh, that's simply the bit of DNA that we design those primers to is 401 base pairs from the beginning of the sequence to the end of the sequence. So we designed our primers so that first base of the primer here is 401 bases away from the first base of the primer over here. So they are literally bookends. They define the ends of the piece of DNA that you're going to be amplifying. And so they were designed so they were 401 base pairs apart, because that's big enough to span exon 1 and see what changed there. So the 401 base pairs comes from what we did when we designed the primers, because we designed them to be 401 base pairs apart, basically. So it's nothing inherent in PCR. It's not like all PCRs will generate a product that size. If you wanted to, you could design them so that they're 1500 base pairs apart. So if you wanted to span 1, 2, and 3, let's say, I don't actually know how far apart they are. I can't remember off the top of my head. But you design primers, one for here and then one for over here, that would then bookend a much larger piece of DNA. Just depends on what you want to amplify and what you're able to amplify too. Because Taq polymerase isn't actually that great of an enzyme. So it'll only do, like 3,000 base pairs, give or take, reliably. Other ones will get up to like 5,000 to 10,000 base pairs. Yeah, for sure you can. I'd be more than happy to do that. Does anybody else have any questions? Da. Cool. So basically that's what we're doing today. What we'll do next week will be, clean up. Actually, no, this Thursday. Sorry, kind of forget what's what sometimes. This Thursday will be all about getting this PCR product cleaned up and submitted for sequencing. So we'll talk about how that happens on Thursday. So the aim of today is generate enough DNA, like physically, that you can actually do something with it. Just the DNA that you extracted from flies is not enough. You might have hundreds, probably thousands of copies of that bit of DNA. But with PCR, you get millions and only the bit of DNA that you want, not all the rest of the genome of the organism that will potentially screw things up. Cool. I think that's pretty much it. If there's anything that on the assignment I didn't cover, which is always possible, but I try not to, feel free to ask me about it on Thursday. Does anybody have any questions about this or the assignment that's due tonight? Oh. All right, cool. Thanks very much, everybody. I shall see you on Thursday. Adam, I'll just be sending you a link so we can meet separately after lab. Cool. All right. Have a great afternoon, everybody. Hope it doesn't rain too much wherever you're at. And I'll see you on Thursday.